Bismillahirrahmanirrahim. I am your course instructor of physiology and my name is Quratr Nazar. Today we are going to start the second topic of our third chapter, and the name of this topic is active transport diffusion. Previously we have discussed about the passive transport diffusion, and today we are going to study about what is active transport diffusion. So basically, active transport diffusion is the movement of substances against the chemical or electrical or electrochemical gradient. It is like swimming against the water tide in a river. It is also called uphill transport diffusion. Active transport requires energy, which is obtained mainly by the breakdown of high energy compounds like adenosine triphosphate. Now, active transport basically differentiate with facilitated diffusion in some ways. And these waves are by carrier protein. Carrier proteins of active transport needs energy, whereas the carrier protein of passive facilitated diffusion does not need any energy. And in active transport, the substances are transported against the concentration or electrical or some electrochemical gradient. But if you can see in the passive facilitated diffusion, you can clearly observe the substances are transported along the concentration gradient or electrochemical or electrical gradients. Now, carrier proteins of active transports are involved in the active transports in two ways. The one way is the uniport system and the second is the symport, which is also called the antiport system. What is uniport system? Uniport is basically defined as the carrier protein that actually carries only one substance in a single direction, and it is called basically uniport. It is also known as uniport pump. Symport or antiport. Basically these two words have the same meanings, but you can say that in one system of symport, the substances move only in the one direction, and in this second system which is called antiport, the substances actually move parallel but in opposite direction. Symport or antiports are the carrier protein that actually transfers two substances at a time. Carrier protein that transports two different substances in the same direction is called symport. That's why I say that in symport, these substances move parallel but in the same direction. And it is also called symport pump. And carrier protein that transports two different substances in opposite direction, it is called antiport, and this antiport system is called antiport pump. Now, the mechanism of active transport. Basically, we can say that when a substance to be transferred across the cell membrane, which is lipid bilayer in nature, comes near the cell, it combines with the carrier protein of the cell membrane and forms substance protein complex, and this complex moves towards the inner surface of the cell. The cell membrane, these substances is released from the carrier proteins. The same carrier proteins then move back to the outer surface of the cell membrane to support another molecule of the substance. So this is how we can easily understand the mechanism of active transport system. Substances which are actively involved in the transportation are in ionic form, and you can say that they can in non-ionic forms. Substances in ionic form are sodium, potassium, calcium and hydrogen. In addition, you can also say that chloride and iodide ions are also in ionic forms. And substances which are non-ionic in nature are glucose, amino acids and urea. Now, previously we have also discussed the types of passive transport diffusion, in which we say that there is one type which is called passive simple diffusion and the other one is the facilitated simple diffusion. Same like the active transport diffusion have some types, and they are basically divided into two categories, and one category is called the primary transport mechanism and the second one is called the secondary active transport. Now, what is primary active transport? Primary active transport is basically a type of transport mechanisms in which the energy is liberated directly from the breakdown of ATP. Now, this is the very clear difference of primary active transport from passive active transport. What happens in the passive active transport? There is no need of energy, but here you can see that in primary active transport, energy required and breakdown of ATP is must for the transportation of substances from the extracellular fluid environment to the internal cellular fluid environment. By this method, the substances like sodium, potassium, calcium, hydrogen and chloride are transported across the cell membrane. Now, the primary active transport of sodium and potassium pump is the best example to understand this system of active transport mechanism. In sodium and potassium ions, basically ions are transported across the cell membrane by means of a common carrier protein, and this is also called sodium-potassium pump system. It is also called sodium-potassium ATPase pump or sodium-potassium ATPase. So you can easily say that sodium-potassium pump is also called sodium-potassium ATPase, is also called sodium-potassium pump. So what this pump actually do? This pump transports sodium from the inside to inside the cell and potassium from outside to inside of the cell. And this pump is present in all the cells of the body. Sodium-potassium pump is basically responsible for the distribution of sodium and potassium ions across the cell membrane of lipid bilayer and the development of the resting membrane potential. So, sodium have some very specific role in developing this resting membrane potential and also maintaining the negative charge inside of the intracellular fluid environment. Now come towards the structure of the sodium-potassium pump. Basically, the carrier protein that constitutes sodium-potassium pump is made up of two protein subunits, an alpha subunit and a beta subunit. And the alpha subunits have a molecular weight of 10,000 and the beta subunits have a molecular weight of 55,000 nanometer. Now, these transportation of sodium and potassium occurs only by the alpha subunit. The beta subunit is a glycoprotein in nature and the function is not actually clear. Alpha subunit of the sodium-potassium pump has got 6 attachment sites, 3 receptors for sodium ions or the inner towards cytoplasm. It means that 3 receptors of the sodium ions are actually located at the inner side of the cytoplasm and surface of the protein molecules. The 2 receptor sites for potassium ions which are located on the outer side, which is also called the extracellular fluid environment of the cell membrane, and basically these are located at the surface of the protein molecule. One site for enzyme adenosine triphosphate which is called ATPase, and it is basically located near the sites for sodium attachments. Now come towards the mechanism of action of sodium and potassium pump. 3 sodium ions from the cells which get attached to the receptor sites of the sodium ions to the inner surface of the carrier protein. 2 potassium ions outside the cell bind to the receptor side of potassium ions which are located on the outer surface of the carrier protein. In this picture, you can clearly see that the binding of sodium and potassium ions to carrier proteins activates in stage 1. And what happens in stage 2? The enzyme ATPase causes breakdown of ATP in the adenosine diphosphate with the release of high energy of phosphate. And what this energy actually do? The energy which is liberated causes some sort of conformational changes in the molecule of the carrier protein. And because of this, the outer surface of the molecule with potassium ions now faces the inner surface of the cell. It means the potassium ions now face, which are actually present in the extracellular environment, they now came in the extracellular environment. And the inner surface of the protein molecules with sodium ions faces the other side of the cell, and this all happening basically occurs in the stage 2. Now dissociation and the release of the ions takes place, so the sodium ions are released outside the extracellular fluid environment and the potassium ions are released inside of the cell, which is called the intracellular fluid environment. Now here you can see the mechanism of sodium-potassium system, which is already explained to you. Now if you can see in figure 1, you can easily see that the 3 sodium ions which are present in the extracellular fluid environment get bindings with the carrier protein, and in stage 2 you can see that ATP enzyme is actually break down into the ADP, and what happens, the extracellular fluid environment, the gate is open, and in stage 2 you can easily see the gate is closed in the extracellular fluid environment, And in stage 4, you can see that these 3 sodium potassium ions actually get released into the extracellular environment. And as a result, the carrier protein opens at the level of extracellular fluid environment, and when these 3 sodium ions get released, 2 potassium ions enters into the carrier protein, they form some conformational changes, and after the attachment they get released in the intracellular fluid environment in stage 5, and this is how this cycle repeats. Electrogenic activity of sodium potassium pump. Basically, sodium potassium pumps moves 3 sodium ions outside the cell and 2 potassium ions inside the cell. Thus, when the pump works once, there is a net loss of 1 positively charged ion from the cell, and the continuous activity of this sodium potassium pump causes reduction in the number of positively charged inside the cell, which is the intracellular fluid environment of the cell, to increase the negativity charge inside the cell. That's why we call that sodium maintains the cell polarity inside the cell. Sodium maintains the negative charge inside the cell. This is called the electrogenic activity of sodium potassium pump. Now, in this picture, you can clearly see the electrogenic activity of the sodium potassium pump. In this picture, you can see that the concentration of sodium in the intracellular fluid environment is lower as compared to the extracellular fluid environment. The reason behind this, in each cycle, 3 sodium ions actually get released outside the cell, and as a result 2 positive potassium ions get inside the cell, and the concentration of these positive potassium ions inside the cell is greater as compared to the concentration which is lower in the extracellular fluid environment. So here you can see that 2 potassium ions are located in the intracellular fluid environment and 3 positive charge ions of sodium are located at the extracellular fluid environment. So if you compare these charge, which is 3/2, you can easily say that the positive charge in the intracellular fluid environment are lower as compared to the extracellular fluid environment, and this is the reason that these negative charge ions are present inside the cell membrane, which is called the intracellular fluid environment. Now there is an example of the transport of calcium ions. This is also occur in the active transport mechanism of cell. What happens in this? Calcium is actually actively transported from inside to outside the cell by calcium pump. Similarly, like we can say sodium potassium pump, when the transport of calcium ions occurs between the lipid bilayer, we can easily say it that now the pump is called calcium pump. Calcium pump is operated by a separate carrier protein. Energy is obtained from ATP by the catalytic activity of ATPase. Calcium pumps are also present in some organelles of the cell, such as sarcoplasmic reticulum in the muscle and the mitochondria of all the cells. These pumps basically move calcium in the organelles. Now there is another example of active transport mechanism, which is transport of hydrogen ions. And the system of the transportation is same as we previously discussed, like transportation of calcium ions and transportation of sodium and potassium ions. What happens in the transportation of hydrogen ions? Hydrogen ion is actively transported across the cell membrane by the carrier protein, which is called the hydrogen pump. It also contains energy from ATP by the activity of ATP. The hydrogen pump that are present in 2 important organs have some functional significance. In the picture, you can clearly see that the pump is present in the stomach and in the kidney. Hydrogen pump is parietal in stomach. Hydrogen pump is present in the parietal cells of the gastric glands, and they are involved in the formation of hydrochloric acid. And in the kidney, hydrogen pumps basically present in the epithelial cells of the distal ovulated tubules, and collecting ducts are involved in the secretion of hydrogen ions from blood into the urine. Now this is all about the primary active transport mechanism. Come towards the second type of active transport mechanism, which is called the secondary active transport mechanism. Basically, the secondary active transport mechanism, the transport of substances with sodium ions by means of a common carrier protein. When sodium is transported by carrier protein another substance is also transported by the same protein simultaneously. You can also say that the another substance is actually using the movement of sodium molecules, and it actually what does, it takes the association of sodium substances and then it moves in the compartment with the help of this sodium, either in the same direction of the sodium movement or in the opposite direction. It doesn't matter. The movement of the substance also in the unipode direction and also in the antipode direction. Thus, the transport of sodium is coupled with the transport of another substance. Now, basically the secondary active transport is also divided into two categories. One is called the co-transport mechanism and the second one is the counter-transport system. Sodium co-transport mechanism. In sodium co-transport, basically the substance moves along with the sodium and another substance is transported by carrier protein, which is called symport. Energy for movement of sodium is actually obtained by the breakdown of ATP molecules. Energy for movement of sodium is obtained by the breakdown of ATP molecule, and the energy released by the movement of sodium is utilized for movement of another substance, and substance carried by this sodium co-transport are normally glucose, amino acids, chloride, iodine, ions and urate. Now the carrier proteins for the sodium co-transport. Carrier proteins which are involved in the sodium co-transport has two receptor sites on the outer surface of the cell membrane. Among the two sites, one is for the binding of the sodium and the another side is for the binding of the other substance. Sodium co-transport of glucose ion. In the diagram, you can clearly see that the postulated mechanism for sodium co-transport of glucose is mentioned. In carrier protein, you can see that carrier protein has two receptor sites on the other surface. Among the two sides, one side is for the binding of sodium and the another side is for the binding of another substance, and these both molecules came in the direction, either this direction is antipode and it may be sympode, but they can move easily along with the help of sodium ions. Now let us understand this sodium co-transport mechanism with the help of example, and for that I would like to add the sodium co-transport of glucose system. It can be explained like one sodium ion and one glucose molecule from extracellular fluid environment bind with the receptor sites of carrier protein of the cell membrane. Now the carrier protein is basically activated. It causes some conformational changes in the carrier protein so that the sodium and glucose are released in the cell. Sodium co-transport of glucose occurs during the absorption of glucose from the intestine and reabsorption of glucose from the renal tubule of kidneys. You can see at first stage, which is the A stage, sodium is attaching with the carrier protein, and along with this sodium, glucose substance is attached. And what happens when these both substances get attached with the carrier protein? They are released in the extracellular fluid environment at B compartment. And in the last, you can easily see that these two substances get released in the intracellular fluid environment. Now take another example of sodium co-transport of amino acids. Carrier proteins for the transport of amino acids are different from the carrier proteins for the transport of glucose. These carrier proteins vary with different substances. For the transport of amino acids, basically there are 5 sets of carrier proteins in the cell membrane. Each one carries different amino acids depending upon the molecular weight of the amino acids. Sodium co-transport of amino acids also occur during the absorption of amino acids from the intestine and reabsorption from renal tubules. Now the another type of system is called the sodium counter-transport. Previously we have discussed sodium co-transport, and now I am going to explain the sodium counter-transport system. In sodium counter-transport, basically it is the process by which the substance are transported across the cell membrane in exchange of sodium ions by carrier proteins which are called antipode directions. And various counter-transport systems are sodium-calcium counter-transport, In this, sodium and calcium ions moves in the opposite direction with the help of a carrier protein. This type of transport of sodium and calcium ions is present in all the cells. Sodium-hydrogen countertransport. In this system, hydrogen ions are exchanged for sodium ions. It means when sodium ions move from one compartment to another compartment, as a result, hydrogen ions replace this, and this occurs in the renal tubular cells. The sodium ions move from tubular lumen into the tubular cells, and the hydrogen ions move from tubular cells into the lumen. It means that sodium and hydrogen basically replace their settings. And in this picture, you can see that sodium countertransport, stage 1, sodium Na positive from extracellular fluid environment and hydrogen H positive from intracellular fluid environment binds with the carrier protein. And what happens in the stage 2? Some conformational changes occur in the carrier protein, and in the last stage, sodium enters in the intracellular fluid environment and hydrogen enters in the extracellular fluid environment. It means the sodium and hydrogen basically replace their positions. Other counter-transport systems are sodium-magnesium, sodium-potassium, calcium-magnesium, calcium-potassium, and they all are called sodium-magnesium countertransport, sodium-potassium countertransport, calcium-magnesium countertransport, calcium-potassium countertransport, and side by side chloride bicarbonate countertransport and chloride sulfate countertransport also occur in these countertransport systems. Now here you can see the two pictures basically represents the sodium co-transport mechanisms and sodium countertransport mechanism. First of all, I am going to explain the co-transport of sodium. Here you can see that sodium is basically help chloride ions to move in the intracellular fluid environment in the co-transport mechanism. And likewise, glucose and amino acids also came in the intracellular fluid environment with the help of sodium ions. And in the counter-transport systems, you can see that ions are exchanged. For example, sodium ion is exchanging with the hydrogen ions, and the second example of the counter-transport is sodium and calcium. Here you can see that sodium ion is basically replacing its space with the calcium ions. Sodium ions enter in the intracellular fluid environment, and as a result, calcium ions comes out from the intracellular fluid environment to the extracellular fluid environment. Now there are some special types of active transport systems. In addition to the primary and secondary active transport systems, there are some special categories of active transport systems which are generally called the vesicular transport mechanisms. Special categories of active transport systems include endocytosis, exocytosis, and transcytosis. First of all, we need to understand that what is endocytosis. Basically, endocytosis is defined as a transport mechanism by which the macromolecules enter the cell. Macromolecules, substance with larger molecules who have larger diameter, cannot pass through the cell membrane either by active or by passive transport mechanism. So, such substances adopted the transportation in the cell by this process of endocytosis. Endocytosis is further categorized into three types, and these are pinocytosis, phagocytosis, receptor-mediated endocytosis. What is pinocytosis? In pinocytosis, basically it is a process by which macromolecules like bacteria and other antigens are taken into the cell. It is otherwise called the cell drinking. Mechanism of pinocytosis involves the following events, and these events basically include some steps. In step 1, macromolecules in the form of droplets or fluids bind to the other side of the cell membrane. After attachment, the cell membrane invaginates around the droplets, and these droplets basically engulfed by the membrane, And you can see at the stage 4 that these engulfed droplets are basically converted into the vesicle and vacuoles, which are called endosomes. After the formation of the endosomes, these endosomes travel into the anterior layer of the cell and form the primary lysosome. And this primary lysosome is present in the cytoplasm. It fuses with endosome and forms secondary lysosomes. Now the hydrolytic enzymes which are present in the secondary lysosomes are activated, result in digestion and degradations of the endosomal contents. Phagocytosis, which is the second type of active transport. What happens in phagocytosis? Basically, phagocytosis is the process by which the particle larger than the macromolecules are engulfed into the cells. It means that it is the advanced level of pinocytosis. It is also called the cell-eating, large bacteria, larger antigens and other large foreign bodies which are taken inside the cell by means of phagocytosis. Only few cells in the body like neutrophils, monocytes and the tissue macrophages show phagocytosis. Among these cells, the phagocytosis are the largest phagocyte cells. Now the mechanism of phagocytosis. If we are discussing the mechanism of phagocytosis, the bacteria or foreign bodies which need to be entered in the body, first the phagocytic cells send cytoplasmic extension, which is called the pseudopodium, around the bacteria or foreign body. Then these particles are engulfed and are converted in the endosome-like vacuole. This vacuole is basically very large in size and it is usually called phagosome. Phagosome travels into the interior cell. Primary lysosomes fuses with the phagosome and forms secondary lysosome. Hydrolytic enzymes then act on the secondary lysosomes and activates the resulting, which results in the digestion and degradation of the phagosomal contents. Now the third type of the active transport mechanism is the receptor-mediated endocytosis. What happens in this? Receptor-mediated endocytosis is the transport of macromolecules with the help of a receptor protein. Surface of the cell membrane has some pits which contain a receptor protein called clathrin. Together with the receptor protein which is called clathrin, each pit is called receptor-coated pit. These receptor-coated pits are involved in the receptor-mediated endocytosis. By this picture, you can understand the mechanism of receptor-mediated endocytosis. Receptor-mediated endocytosis is actually induced by substances which are called ligands. Ligand molecules approach the cell and bind to the receptors in the coated pits and form ligand-receptor complex at the level of the cell membrane, which is also called lipid bilayer. Now these ligand receptors complex get aggregated in the coated pits. Then the pit is detached from the cell membrane and becomes the coated vesicle. This coated vesicle forms the endosome. And then what does endosome do? This endosome basically travels in the anterior of the cell. Primary lysosome in the cytoplasm fuse with endosome and form secondary lysosomes. Now the hydrolytic enzymes which are actually present in secondary lysosomes are activated, resulting in release of ligands in the cytoplasmic complex. And these receptors may move to new pit of the cell membrane and repeat this process when this process is required or when this process is needed. Now these receptor-mediated endocytosis play a very important role in the transport of several types of macromolecules in the cell. The first one at the level of hormones, growth hormones, thyroid stimulating hormone, luteinizing hormone, prolactin, insulin, glucagon, calcitonin and many other hormones basically play an important role in the transport of several types of macromolecules in the cell. The second one is the lipid, and cholesterol, low density lipoproteins are involved in lipid categories. The third one is the growth factors, which involve the nerve growth factors, epidermal growth factors, Platelets drive growth factors and interferon. The fourth one is the toxins and bacteria. Cholera toxins, diphtheria toxins, monas toxins, racins, and concanevalin A type of toxins. The other one is the viruses like Rose, sarcoma virus, Semiliki, forest virus, vesicular stomatitis virus, and adenovirus. The another one is the transport proteins like transferrin and transcobalamins. And the last one is the antibodies, and these antibodies are present in the body in the form of IgE, polymeric IgE, and maternal IgE. Some of the receptor-coated pits in cell membrane are coated with another proteins which are called cavalin instead of clathrin. Cavalin-coated pits are concerned with the transport of vitamins into the cells. Now, what is exocytosis? In exocytosis, basically it is the process by which the substances are expelled from the cells. In this process, the substance are excreted from cell without passing through the cell membrane. This is the reverse of endocytosis. In exocytosis, the release of secretory substances from cells occur. Secretory substances of a cell are stored in the form of secretory vesicles in the cytoplasm. When required, the vesicles actually approach the cell membrane and get fused with the cell membrane. Later, the content of the vesicles are released out of the cell as shown in the diagram. Now, what is the role of calcium in exocytosis? Basically, calcium ions play a very important role during the release of some secretory substances such as neurotransmitters. These calcium ions basically enter the cell and cause exocytosis. However, the exact mechanism of exocytosis is not clear, but these calcium ions play a vital role in the process of exocytosis. Now, the last one is the transcytosis. What happens in the transcytosis? Transcytosis is a transport mechanism in which an extracellular macromolecule enters through one side of the cell and it migrates across the cytoplasm of the cell, and then it exits through the other side of the cell. Now, the mechanism of the transcytosis. Cell encloses the extracellular substances by invagination of the cell membrane to form a vesicle. Vesicles then move across the cell and thrown out through opposite cell membranes by means of exocytosis. Transcytosis involves the receptor-coated pits as in receptor-mediated endocytosis. These receptor-mediated protein coated the pits in the process of cavalin, not clathrin. Transcytosis is also called vesicle trafficking or cytopamsis. Transcytosis play a very important role in selectively transporting of substances between two environments across the cell without any distinct change in the composition of these environments. Example of this type of transport is the movement of proteins from capillary blood into the interstitial fluid across the endothelial cells of the capillary. Many pathogens like HIV, which is called human immunodeficiency virus, also transported by this type of mechanism. Now, what is molecular motors? Molecular motors are actually the protein-based molecular machines that perform intracellular movement in response to some specific stimuli. What is the function of these molecular motors? They help in the transport of synaptic vesicle containing neurotransmitters from the nerve cell body to synaptic terminal. The second function of the molecular motors is help in the role in cell division, that is mitosis and meiosis, by pulling the chromosomes. And third one is the transport of viruses and toxins to the interior of the cell for its own determinants. Now, there are some different types of molecular motors which are present inside and outside of the cell, and they control the basic functions. And these motors are kinesins, dynesins, and myosin. What kinesin does? Kinesin transports substances by moving over the microtubules. Each kinesin molecule has two types of heads and a tail portion. One of the head hydrolyzes ATP to obtain energy. By utilizing this energy, the other head swings continuously, causing movement of the whole kinesin molecule. As you can see in the picture, end portion of this cell, end portion of this tail basically carries the cargo, which is the substance to be transported. Kinesin is responsible for anterograde transport, which is the transport of substance towards the positive end of the microtubule. Function of the dynein is almost similar to the kinesins and transports the substances by moving over the microtubules, but it is responsible for retrograde transport, which is the transport of substances through the negative end of the microtubule. Here you can see the picture of the kinesin and dynein, and you can see that these two substances help in the movement of molecules with the help of this cargo. Basically, cargo is the substance which kinesin and dynein needs to move. Either they may be in the positive side and they can also at the negative end. Myosin. Myosin helps in the transportation of substance by moving over microfilaments. Myosins are classified into 18 types according to the amino acid sequence. However, myosin 2 and 5 are functionally significant. Myosin 2 is involved in muscle contraction. Myosin 5 is basically involved in the transport of vesicles. Now the applied physiology of abnormalities of sodium-potassium pumps. Abnormalities in the number of functions of sodium-potassium pumps are associated with several pathologies, and these pathological conditions are reduction in either the number or concentration of sodium-potassium pump in microcardium and is associated with cardiac failure. It means that the body needs sodium-potassium pump at regular interval. If any ambiguity or any abnormality occurs in this pump, then body may be shifted in the cardiac failure. And in the 2, you can see that excess reabsorption of sodium in renal tubules is associated with hypertension. Channelopathies or ion channel diseases also occur. Channelopathies or ion channel diseases are caused by the mutation in genes that encodes the ion channels. Sodium channel disease basically causes the dysfunction of sodium channels, which leads to muscle spasm, which is called Liddle syndrome. This syndrome is basically occurs due to the dysfunction of sodium channels in kidney, resulting in increased osmotic pressure in the blood and causes hypertension. What happens when potassium channel disease occurs? Potassium channel dysfunction causes disorders of heart, which leads to cardiac failure, inherited deafness, and epileptic seizures in the newborn. Chloride channel disease. What happens if the disease occurs at the level of the chloride channels? Dysfunctions of the chloride channels results in the formation of renal stones and cystic fibrosis. Cystic fibrosis is a generalized disorder which affects the functions of many organs such as lungs due to excessive mucus, exocrine glands like pancreas, biliary systems, and immune system. Now this is all about the 3rd chapter of physiology, and in this chapter you covered the active transport mechanism, passive transport mechanisms, their different types, and in the end you covered the basic applied physiology, the abnormalities and the diseases which occur due to the differentiation or any mutation occurs at the level of this active and passive transport system. I hope you all understand well. Thank you.